Hello, AP Bio. Welcome to our video lecture for Chapter 16, Development, Stem Cells, and Cancer. So for this chapter, I picked a picture of Jack. In this picture, he's right around 1, and I love this picture because, it shows some development going on. You can see he's reading a book. This is one of those little books from Chick-fil-A. The book, however, is upside down, so the development's not quite there yet. But, he's getting there. So this chapter is a bit of a hodgepodge chapter. It picks 3 topics: development, stem cells, and cancer, which are related for sure, but are kind of distinct in their own right. So the development part, so we've said many times in this class that you were once a zygote. You began life as a zygote, and you've grown into an organism of millions or trillions of cells that are differentiated. Have skin cells and brain cells and muscle cells and blood cells. How that process works is what development is talking about. So to study this, we use model organisms, things like fruit flies, to try and figure out how you can go from a zygote with all of your genes to a multi-celled organism with all of your genes in every cell, but where only certain genes are expressed at a time. Again, this slide just shows that actually the last point on the slide, these terms cell division, cell differentiation, and morphogenesis. Obviously, cell division, you know what that means. Those are terms that we need to go through. But the idea here is that how genes are expressed as you go from a zygote to a multi-celled adult changes. Okay, this picture just shows fertilized frog eggs becoming a tadpole. So obviously, this is development. This is cell differentiation, because you're going from one cell to multi-cells that do different things. So cell differentiation, this might be a new word, but it's a very simple concept. It's just the idea of a cell becoming specialized. So a zygote is not specialized. Stem cells, not specialized. A skin cell is specialized. Sometimes in class, I like to use the term growing up, because it's when a cell grows up, when a cell becomes what it's supposed to be. And once a cell is differentiated, typically it can become more cells of that type, but it can't go back to being undifferentiated. Once a stem cell becomes a blood cell, it can't go back to being a stem cell, and it can't become, say, a nerve cell. Morphogenesis is just the term for the processes that an organism goes through to get its physical shape, to grow up. In differential gene expression, that just means expressing genes differently in different types of cells or different times during the cell's development. So cytoplasmic determinants. So this is a super important concept. So these are determinants in the cytoplasm, usually RNA or maybe proteins, and they're found in the cytoplasm of the egg cell, and they're important not just for what they do, but for where they are. So in terms of fertilization, the sperm really just contributes 23 chromosomes, and that's really about it. The egg obviously contributes 23 chromosomes, but the egg does way more in terms of developing zygote than the sperm does. For example, all of the cytoplasm in the zygote came from the egg. The sperm contributes no cytoplasm. So all the mitochondria that you have, going back to when you were a zygote, those came from your mother. So you inherit mitochondria maternally. Remember, there are genes in mitochondria. If you go back to chapter 7, I believe, or maybe chapter 4, I can't remember. Maybe both. But your mitochondria have genes. So the genes in your mitochondria came from your mother, not from your father. Mitochondrial DNA is inherited maternally. And actually, you can get mitochondrial DNA from crime scenes and forensics. So the rules of inheritance are a little bit different because they come from the mother. Now look at this picture here. Do I have this bigger? No, I don't. So here I have an egg. I have orange circles and green triangles. Say those are RNAs. The egg is already made in advance of fertilization. The circles are on the left, the triangles are on the right. So when that cell divides, those become partitioned. So maybe the circles are RNAs that help form the head of the organism, and the triangles help form the tail of the organism. Where those molecules are physically makes the difference. If I move them around, you mess up the development, which we'll discuss later in this chapter. Induction is just the idea. So here I have a cell. giving off some kind of signal molecules, and they affect the ones nearby. So induction, or to induce, is just when you have interactions between cells that are literally in the same place. Like if one cell gives off a hormone, it's going to affect the cells closer to it more than the ones further away in an embryo when there's no, when there's no blood. If you have blood, if hormones get into the blood, if they go everywhere. But this is in like an early embryo, like the 32 or 64 cell stage. So these were determination and differentiation. So this is an important distinction. So we've already said differentiation is when a cell grows up, when a cell becomes specialized. Determination is when the cell's fate has already been determined, even though it might not have grown up yet. In this last point, determination precedes differentiation. A cell's fate is locked in before it actually becomes what it's going to be. So say a stem cell gets tagged to become a muscle cell. It's going to be a muscle cell. It's been determined, but it's not a muscle cell yet. So you determine the fate before you differentiate the cell into what it's going to be. So this is an interesting slide. This looks complicated, but it's really not. Myosin is a family of proteins found in muscle cells. So we're talking about the determination and the differentiation of a stem cell, a precursor cell, into a muscle cell. So you have tons of genes that make muscle proteins, right? And here, they're turned off. In a stem cell, stem cells don't have muscles, right? It's just a cell. And the muscle cells or the muscle genes are turned off. Well, here's a master regulatory gene that codes, this is a gene that codes for a transcription factor that will simultaneously turn all of these genes on. So once a stem cell becomes determined to be a myoblast, which is a kind of muscle cell, you turn the regulatory gene on, and that bad boy turns on all the muscle genes. So at this point, this cell has grown up, or its fate has been determined. It will grow up to be a muscle cell. And you didn't turn all these genes on separately. You turned them all on at once with the regulatory gene. If there was a mutation in the regulatory gene, that would be game over. The cell would likely die. It definitely would not become a muscle cell. Okay, apoptosis. You've heard this word before. This just means programmed cell death. So there's many reasons why a cell might be designed to eventually die. When cells become infected, there's ways that it can sort of wave a white flag to trigger an immune response to come kill it because it's infected. So it sort of wants to die to prevent the infection from spreading. Cancer cells, when cells become cancers, they can sometimes initiate apoptosis. We discussed earlier, so this picture, this is some kind of vertebrate. I'm not sure what it is. The embryo of some kind of vertebrate. And you can see this is like the hand, and the space between digits is sort of being carved out, right? The cells in those spaces are going through apoptosis, going through programmed cell death to die, and that's a normal part of development. A tadpole losing its tail is another example. The DNA is broken up, organelles become fragmented, and the cell eventually just dies. So switching gears a little, pattern formation. So here I have some kind of insect, say it's a fruit fly. So there are genes that code for the insect's head, thorax, and abdomen, right? Those are genes that do that. Well, there's also genes, excuse me, there's also genes that code for where to put the head, the thorax, and the abdomen, and those are called pattern formation genes. Oftentimes, the cytoplasmic determinants in the egg cell help dictate, where the head goes, where the thorax goes, where the abdomen goes. Notice the legs of this fly are coming out of the thorax. So there are genes for legs, but there's also genes to say, where do I put the legs in the thorax? Those are called pattern formation genes. One class of these are called homeotic genes. These were discovered by Edward Lewis. He got the Nobel Prize in 1996. Look at this picture. This is a fruit fly with the antenna, the eyes. This is normal. This fruit fly has legs growing out where the antenna should be. So what he did, he didn't change the gene for legs. The gene for legs is the same. He just changed what are called homeotic genes, which are pattern formation genes. The legs wound up in the wrong place. Okay. Axis establishment. So maternal effect genes are just maternal effect refers to the egg, right? These are genes that are coding for the cytoplasmic determinants that help establish body polarity, like where's the head, where's the thorax, where's the abdomen. One type is called the bicoid gene. You don't need to know specifically about the bicoid gene, but here I have the embryo or the larva, sorry, of some kind of insect. T is thoracic, A is abdomen. You see the tail, this is normal. Here, the abdominal segments 8, 7, 6, 7, 8. Here I messed with a bicoid gene. So, it doesn't change the abdominal segments, but it changes where they are, okay? So just file away the fact that not every gene codes necessarily for like legs. Some genes code for proteins that determine where the legs go. Okay, cool. This is an experiment. So the bicoid DNA that we mentioned, actually it wasn't DNA, it was RNA that we mentioned. They've dyed it blue. You can see where it is. And the end that had the bicoid RNA became the anterior or the front end. And if what he did, if I moved the RNA to this end, then this end becomes the front end. If I have it half and half, both ends become the front end, kind of like something like here. Although in this picture, both ends became the tail end. Okay. You can read this yourself. Just the point of this research was it shows how important the egg is, the unfertilized egg is, toward the development of the embryo. Okay, switching gears. Let's discuss cloning for a second. So cloning is the idea of taking things that reproduce sexually, but reproducing them only requiring one cell with no meiosis or fertilization. The way that cloning works, so the word totipotent is something you can describe as stem cells, that they can become any type of cell. So look at this picture. Do I have this bigger? No, I don't. So I have an egg cell and I zapped it with UV radiation, and basically you enucleated it. So take this egg cell and get rid of the nucleus, all right? Then either replace it with a nucleus from a differentiated cell. This shows an intestinal cell of a tadpole, or replace it with a nucleus from a cell of an embryo. So I take the donor egg, get rid of its nucleus, and put in a different nucleus that's already diploid. Like the nucleus of this egg was haploid. We'll replace it with a nucleus that's diploid, and if it works, you're going to get a zygote that is genetically the same as either the embryo or the tadpole. This obviously is difficult to do because there's a lot of reasons why this wouldn't work. It does work. We can clone lots of things, and obviously you didn't need egg or sperm. You didn't need, well, actually you did need a donor egg, but you didn't require fertilization. So just so we're clear, when this thing develops, is it identical to the creature that gave the donor egg? No, it's identical to the critter that donated the nucleus. Okay, so just quick history lesson. So in 1997, the first mammal, I believe, was cloned in Scotland, and it was a female sheep named Dolly. You might have heard of Dolly the sheep before. This is Dolly. This is Dolly taxidermy. You can go visit her in a museum if you want. And this was huge. I was in high school when it happened. Actually, I was 17 years old, so I was about your age when this happened. It made the news a lot. Dolly was sort of famous, not just because she was the first cloned mammal, but Dolly, Dolly went on to have baby sheep the natural way. So that showed that cloned organisms can reproduce normally. Dolly did die earlier than most sheep do, and she had arthritis. And for a while, people were convinced that cloned organisms, don't live as long, or their cells aren't as robust. But if you read about Dolly, the flock that she lived in, a lot of those sheep had arthritis, sheep that were not cloned, and a lot of them died prematurely. I think there was some kind of, I don't know if it was a virus, some kind of pathogen. So her premature death was probably not due to the fact that she was cloned. It was probably due to some kind of issue with the flock itself. The researchers that cloned Dolly, so what they did, so they took the donor egg from a different sheep. The donor nucleus came from a mammary cell from a different sheep. So when Dolly was cloned, she was identical genetically to the donor nucleus, the sheep that gave the donor nucleus, which was from a mammary gland. And actually, fun fact, the researchers named her Dolly after Dolly Parton. You know, I'm a huge Dolly Parton fan, so shout out to Dolly Parton. And they picked the name Dolly because the donor nucleus was from a mammary cell, or a cell from a mammary gland. And if you can't put together why, it's supposed to be funny, why mammary cells is associated with Dolly Parton. If you don't quite get that connection, ask me in class and I'll explain it to you. But they're being tongue-in-cheek. Those Scots, they have a clever sense of humor, right? Since then, we've cloned lots of mammals. Cloned mammals don't always look or behave just like the parents, the whole nature versus nurture debate. If I don't feed them as well, they won't grow as big. So they're not identical in appearance necessarily, but genetically they are. And most cloning experiments don't work. Only a small percentage actually are successful. So a stem cell, like we said, these are unspecialized or undifferentiated cells that can become, if they're embryonic stem cells, they can become anything. The adults do produce stem cells. For example, in your bone marrow, there are stem cells. Those stem cells don't produce anything. They produce different kinds of blood cells. So embryonic stem cells, those were blastocysts. That's just a stage of development of an embryo. They can become anything. Adult stem cells can become more than one thing. This just shows embryonic stem cells can become anything. Bone marrow stem cells can become different kinds of blood cell. The word pluripotent is just another word meaning cells that can become pretty much anything. There are ways of reprogramming differentiated cells to become stem cells using retroviruses. I'm not sure how you do that, but it does happen. Obviously, there are some serious ethical issues. If the only way to get stem cells is from an embryo, you have to destroy the embryo, which ethically that's questionable, right? If there's a way of reprogramming adult cells to become stem cells, there really aren't any ethical situations there. Few people would argue with that, like they debate about destroying human embryos, which is definitely a no-no, right? Okay, last topic in this chapter. So cancer. Okay, this gets pretty deep, so let's just hit some vocabulary. So, you know, a cancer doctor is called an oncologist, right? So everyone has genes called, actually, back up a second. Oncogenes are what are the name that we give genes that can lead to cancer. Everyone has what are called proto-oncogenes. Proto-oncogenes are normal genes, we all have them, that stimulate cell division. So, you know, you get a cut, those skin cells go through mitosis. You get sick, you need more white blood cells. If you grow, you need more bone cells, right? Proto-oncogenes are normal. If we don't have them, then you'd still be a zygote, right? Proto-oncogenes, though, can mutate into oncogenes, which can cause cancer. If you look at this diagram, here I have it bigger. So here's a proto-oncogene where there was a mutation or a translocation, and I changed the promoter. And this new promoter loves polymerase, so this gets expressed too much. Here, the gene itself got amplified, it got duplicated, so I get too much of this growth factor. Here, I mutated the gene itself, or part of a control element of the gene, and I made growth factor that I either made too much of it, or I made growth factor that was like super growth factor. So for whatever reason, your normal proto-oncogenes, which again are normal, you have to have them, become way turned on. The analogy you can use is a car, where proto-oncogenes that become oncogenes are like taking the gas pedal and hitting it to the floor, all right? A gas pedal makes you go faster. So mutating proto-oncogenes makes those cells go through the cell cycle way too much. So that's one family of cancer-causing genes. The other family are called tumor-suppressor genes. Tumor-suppressor genes are actually good. We want them unless they're mutated. They are genes, remember the whole idea of the G1 and the M and the G2 checkpoints, things that stop cell division? These are genes that code for proteins that slow the cell cycle down. So in our car analogy, they're sort of like the brake pedal of a car, and they put the brakes on the cell cycle. If I mutate tumor suppressor genes, if you can't hit the brakes, you can't slow down, right? These genes might make proteins that might repair damaged DNA, which is good. They might control cell adhesion, Remember how we said that cells stick to stuff, but cancer cells don't? So a cancer cell that doesn't stick to its surface or doesn't stick to the organ it belongs to would become a cancer cell that metastasizes. Or they might be making proteins that inhibit the cell cycle. So think about proto-oncogenes and tumor suppressor genes as normal things that either speed up, gas pedal, slow down, brake pedal the cell cycle. If I mutate them, either you can't slow down or you go too fast. I want to go through two examples. If these were on the AP exam, the AP exam would sort of give you some background. You don't really need to memorize the RAS proto-oncogene and the p53 tumor suppressor gene, but you should be familiar with them if you see it again. So the RAS gene, let me go ahead and I'll come back. Do you remember from chapter 3 or 4, whatever, when we discussed signal transduction, and I said this was very important that we would come back to it later? So quick review. So signal transduction was the idea that you have some kind of receptor protein in the cell membrane. A signaling molecule, like a hormone, binds to it, its shape changes, and it turns on some kind of cascade pathway. Remember there was one called the G protein, where when this thing bound, it turned on a G protein, and then it turned on that whole protein kinase cascade thing through the cytoplasm, and how this binding once could turn on this 1,000,000 times. And the response would be turning a gene on or off, okay? Just refresh your memory that exists. So here I have in the RAS proto-oncogene, this top one is normal. A growth factor binds to the receptor. It activates the G protein, which is the RAS protein. It's a type of G protein. And it turns on the protein kinases, which turn on a gene in the nucleus, which is a transcription factor that turns on the cell cycle. Okay, this is normal, all right? If I mutate that G protein and I form a RAS protein that is like super always turned on, and I like amplify this big time, you overexpress that transcription factor and the cell divides way too much. So the RAS proto-oncogene is normal, we need it. If I mutate it, you call it an oncogene, and that can lead to cancer, okay? Different example, the p53 tumor suppressor gene is a gene that prevents the cell from passing on mutations when DNA gets damaged. So here I shine UV light on a cell. There's DNA damage. That signal, however the cell does it, turns on a kinase cascade, which turns on the active form of p53, which is a protein that binds to DNA and stops the cell cycle. So the cell cycle got stopped because of the UV radiation in the first step. Now, if I mutate the tumor suppressor gene, the p53 gene, and it's a different shape, you know, all this gets turned on, but it's the wrong shape, so it doesn't turn off the cell cycle. So what would happen if I had this down here and I had this down here? That's hitting the gas and breaking the brake pedal, like breaking it as it doesn't work anymore. That's going to lead to cancer. All right. And this deal, there are many different types of genes that are proto-oncogenes or tumor suppressor genes. The multi-step model. So I'm going to just blow up this picture right here. So usually you need to have a mutation in multiple proto-oncogenes or tumor suppressor genes or both. So this is showing this in colon cancer. So one tumor suppressor gene got mutated, so I get cells growing too much. That's a small polyp in your large intestine. That's not good, but it hasn't spread, so it could be worse. Then I'm gonna mutate the RAS proto-oncogene, so now they get expressed more. I'm gonna turn off another tumor suppressor gene. The thing keeps growing. Turn off another tumor suppressor gene. Maybe here I turn off the gene that makes the cells stick together. The cells leave, they get into your blood and it spreads. So oftentimes you need to have multiple oncogenes and tumor suppressor genes that have been mutated to get cancer. Okay. Last thing, what I want to end with. So can you inherit cancer? You can inherit a predisposition to cancer. So the BRCA1 and BRCA2 genes, these two genes are ones you've probably heard about with breast cancer. Everyone has the BRCA1 and BRCA2 genes. Like all women have it, right? The question is, do you have the mutated version, which can lead to skin cancer or lead to breast cancer? And you can do genetic testing to see if you have a mutant version of the BRCA1 or BRCA2 gene. And let me just clarify, men and women can have the BRCA1. A guy can get breast cancer. Famous story, who was it? Angelina Jolie, her mother died of breast cancer, so she had a test done to see if she had the BRCA1, BRCA2 genes, and she did. She had the mutant versions of at least one of them. So she had an elective double mastectomy where they removed her breast tissue as like a way to prevent, like she didn't get breast cancer, but because it was a likelihood, she took that step to try and prevent it, which is that's a pretty serious step. So can you inherit cancer? You can inherit a predisposition to cancer for sure. Obviously, getting sunburns, carcinogens, smoking cigarettes, X-ray exposure can lead to cancer too. That isn't necessarily inherited. Those are things that happen to you during your lifetime. This last point I want to mention for just a minute. Viruses play a role in about 15% of human cancers. So there's one that we should discuss in a little bit of detail, and that's HPV. HPV stands for human papillomavirus, and it is the most common STD in America. Last I checked, I looked this up before I recorded this video, there were 80 million Americans with HPV. That's a lot. It's a virus that is transmitted sexually, and most people that have HPV don't know that they have it. You can have it and have no symptoms at all. It can cause genital warts, which if you get those, there's no way to cure HPV, but there are treatments for genital warts. The big thing with HPV though is that it can cause cancer, particularly later in your life. In women, it can cause cervical cancer. Most cervical cancers are caused by HPV. In men, it can cause throat cancer. This has been studied way more in women than in men because cervical cancer is it's a very deadly kind of cancer. And what scientists found was that the HPV DNA inserts itself into a tumor suppressor gene that's active in cervical cells inside a woman. So the HPV DNA, this was actually discovered, if you've read the book, The Immortal Life of Henrietta Lacks, and learned about HeLa cells. If we haven't discussed HeLa cells in class, remind me because we should discuss this because this is a fascinating story. But it was using the cells of Henrietta Lacks, who was an African-American woman who died in the 50s of cervical cancer. Her cells, whatever, ask me in class, we'll discuss it. But the DNA from HPV inserted itself in a tumor suppressor gene, which disrupted it, which turned it off, or made it inactive. She gets cervical cancer because of the virus. So, like, people always debate, can you catch cancer? Like, directly, no, but you can catch a virus that can lead to cancer for sure. The Gardasil vaccine, many of you might have gotten, it's a vaccine. It's given usually to girls because girls, a guy can't get cervical cancer, but he can get throat cancer from HPV. So maybe guys should get it too. But the idea here is if every girl was immune to HPV, there would be largely no cervical cancer, which again, that's a deadly cancer that usually you get in your 40s or 50s. You might have gotten HPV in your 20s, but you might get the cancer decades later. Okay, yes, if you have any questions about that, feel free to ask me in class. I think that's enough for one chapter. I hope that was helpful, and I will see you guys next time.